Today we will discuss the L-Arabinose Operon, otherwise known as the araBAD or simply AraOperon, that is naturally found in E. coli and allows the bacteria to utilize arabinose as a carbon source. Let's go over the different components of this operon. First, we have a few binding sites: AraO2, AraO1, cap binding site, AraI1, AraI2, and RNA polymerase binding sites. Additionally, we have the regulatory gene AraC and our structural genes AraB, A, and D. The order of these genes is why some refer to this operon as the araBAD operon. The protein components involved in regulation are catabolic activator protein, or CAP, and AraC, as well as RNA polymerase to transcribe the gene. Let's discuss what this operon looks like when active. First, I'd like to mention that in the absence of AraC, RNA polymerase can bind the AraC promoter in order to transcribe AraC mRNA, which is ultimately translated into the AraC protein. In order for there to be high expression of our araBAD proteins, we need to have low glucose in the cell, which will cause an increase in cyclic adenosine monophosphate, or CAMP. CAMP will bind to the CAP proteins, thereby activating them. We also need arabinose, which will be bound by AraC, causing it to dimerize in a specific orientation. In this orientation, the dimerized AraC will bind the DNA elements AraI1 and AraI2. Two of our now activated CAP proteins can also bind the CAP binding site. Additionally, when the AraC is dimerized in this orientation, dimers will also bind to the AraO1 site. This site overlaps with the RNA polymerase binding site for AraC and will prevent RNA polymerase from binding to that promoter, thereby repressing expression of its own gene. This is called autorepression. However, the CAP and AraC dimers can recruit the RNA polymerase to the promoter for AraB, A, and D to trigger transcription of polycistronic araBAD mRNA, which can then be translated into the enzymes required for arabinose catabolism. Now let's look at an inactivated arabinose operon. In this scenario, we have high glucose, which means the absence of CAMP, so our CAP proteins are not activated. We also have low levels of arabinose. AraC will again dimerize, but this time in a different orientation. In this orientation, the dimerized AraC will bind the DNA elements AraI1 and AraO2, which will pull the DNA into a loop. The CAP proteins are not active, so they cannot bind DNA. RNA polymerase is physically prevented from binding the promoter for AraC. And without the CAP proteins, RNA polymerase will not be recruited to the araBAD promoter. But even if it was, RNA polymerase is still physically prevented from binding through DNA looping. Now let's look at some other scenarios. First, let's say we have glucose and arabinose in the media. CAMP is not produced in the presence of glucose, so therefore CAP will be in its inactive form. However, we do have arabinose, so our AraC dimer will not cause DNA looping. In this scenario, our operon is open, so RNA polymerase can bind, but it's not being actively recruited by CAP. This leads to low expression. Now let's say we have neither glucose or arabinose. In this scenario, there is CAMP, so CAP is in its active form. In the absence of arabinose, AraC is still dimerized in such a way that causes DNA looping. So CAP is able to bind to the DNA, but even if it recruits RNA polymerase, it is still prevented from binding to the araBAD promoter, and therefore no mRNA is made. That's all for now. Thanks for watching.